preclinical NHP and large animal studies for AAV-based gene therapy through improved AAV purification and quality control strategies. I'm Tiffany, your moderator for today, and I'm delighted to have each and every one of you with us. Before we begin, let's take a moment to ensure everyone's comfortable and ready to engage in today's discussion. Now, let me introduce our speaker, Dr. Irene Song. Dr. Song is from Packaging Biotech, where she serves as the Senior Director of Global Product since February 2023. Before joining Packaging Biotech, she led the product management team for molecular biology at GenScript. Dr. Song brings a wealth of experience, including her time as a postdoctoral researcher at the Center for Neurodegenerative Disease Research, University of Pennsylvania School of Medicine. There, her focus was on unraveling the mechanism of Parkinson's disease. Her academic journey includes earning a PhD in cell and developmental biology from the University of California. During her research, she dived into a chemical genetic approach, exploring mitochondrial behavior and mammalian cell physiology. Her expertise also extends to apoptosis and investigating the mechanism of dominant optic atrophy. Throughout the presentation, if there's any questions, feel free to submit them to the Ask a Question session, and we'll address them towards the end of this webinar. For any other questions we're not able to answer during the time, we will try to cover them after through email. Now let's get to the heart of today's webinar. I'll turn it over to the speaker for the main presentation. Dr. Song, the floor is yours.
Thanks, Tiffany, for the introduction, and I really appreciate everyone to attend today's webinar. Sorry for the extremely long title, and here today we are going to discuss how we overcome all these challenges in the AAV-based gene therapy when we use an NHP or large animal models, and you know how we overcome these challenges by improving the AAV purification and also have more stringent QC controls. So gene therapy has gained a significant attention in recent years, and unlike the traditional methods involving protein or small molecule delivery, gene therapy directly introduces genetic materials such as DNA or RNA into the target tissues of patients. And additionally, it involves in the ex vivo genetic modification of cells before we can transplant them back into the patients. And one of its standout features is the ability to precisely edit the genome or restore genetic defects, resulting in a treatment that requires very few doses in the patient's lifespan to be effective. And despite the experience up and downs in the recent years, the gene and cell therapy field remains resilient, and it requires shorter development timeline and lower investment compared to other therapeutic approaches, and attracting an increasing number of scientists to dive deeper into this field. And according to the ASGCT 2023 Q3 reports, we still see over 2,000 gene therapies in development, and 560 of them was in the clinical stage from phase 1 to the pre-registration, and there are about 27 gene therapies that are approved that also included the genetically modified cell therapies. Among the various gene delivery methods, AAV stand out as a widely adopted tool. The wild-type AAV genome houses the RAP and CAP gene, encoding the capsid and enzymes that are essential for virus replication. The region between the inverted terminal repeats can be swapped with therapeutic genes, which can form recombinant AAV. To produce recombinant AAV, a transfer plasmid containing the gene of interest with the ITR is co-transfected with an RC plasmid that carrying rapid cap gene, and another helper plasmid that can facilitate virus packaging. And then once the recombinant AAV is formed, the targeted cells can be infected to facilitate gene delivery. AAV boasts several advantages over other viral and non-viral gene delivery methods, primarily due to its superior safety, accessibility, and specificity. AAVs are non-pathogenic and naturally replication deficient. Moreover, in most cases, the AAV genome is non-integrative, acting as an extra chromosome within the nucleus to enable long-term expression of therapeutic genes without messing up the host genome. AAV can infect both dividing and post-mitotic cells, such as neurons. Despite their 4.5 kb genome capacity, they can efficiently pack most single gene expression cassette, and in case of long genes, we can always utilize dual vectors to put the different pieces of gene together. So this versatility positions AAV as an excellent tool for treating neurological diseases, facilitating gene editing for ex vivo cell engineering, and developing vaccines against infectious disease like Ebola, COVID, and Zika virus. One of the standout features of AAV is its diverse tissue tropism, allowing it to specifically infect certain organs, tissues, or cell types. And this capability can be harnessed through natural characteristics or engineered capsids to meet specific needs. Thus, AAV-based gene therapy minimizes the required dosage and reduce adverse effects on unwanted tissue or organs. On the right, you can find a few examples of AAV-based gene therapy approved in recent decades, including Glybera, that was approved in 2012, and Hamgenix, the latest ones that being approved in 2022. And in general, AAV-based gene therapy development can be divided into several different stages. The first stage we call basic biology, which mostly is involved in the target identification. And during that step, people will start to do the genotyping and to phenotype prediction, and sometimes do the high throughput screening to identify the target gene, and also do some functional assays to make some of a proof of concept. The second stage is therapeutic therapy development, and in most of the cases, people will engineer the viral vector optimization. They will find out the best expression cassette, the code optimization for the therapeutic genes, and also they will do some of the in vitro validations, which involves the cell culture, and also do some in vitro toxicology study. The third stage, which I would like to emphasize most, is the pre-IND stage. During that stage, a lot of animal studies was involved. So people will either to establish the animal disease models, or they will do a lot of in vivo efficacy assays or in vivo toxicology studies, and also to study the biodistribution of where the AAV virus will go, to make sure that they will gain enough data for their IND filing. And also during that stage, because they would like to prepare to enter the clinical stage, they will also do a lot of process development and scale up production. They will also do the assay qualification as well as the starting material generation. And once this steps was successfully conducted and approved for the IND, they will be ready to get into the clinical stage and do the phase 1 to 3 clinical studies and get ready for the commercialization. So during pre-IND stage, different animals could be used. The most popular is mice, which because they are always like low cost, they can they can generate results very fast and easy to create disease models compared to other larger animals. However, more and more studies tend to switch to non-human primates, especially if you are studying neurological diseases or respiratory diseases. In some cases, when you need to study muscular or circulatory systems, porcine is also a very good system. So the reason why more and more people start to switch into non-human primates model is because non-human primates has several advantages, especially over mice. Um, also for the mice, because there are a lot of cases, not all the results were transferable to human system, and non-human primates are much closer to the human. For example, like on the anatomical aspects, um, in these the monkeys will have expansions of their neocortex, have high order abilities and social complexities, and also they're easy to control and image neuron circuits in the living NHPs, especially for the central nerve system and the peripheral nerve system. They share very similar physiology with human beings, so transduction and other aspects of the vector will be able to easily evaluate it in the non-human primates, and the pathological process in the neurological diseases are much similar in non-human primates compared to human. And also, they will enable the major advantages in pharmaceutical and vaccine development. Um, Another aspect that makes it have an advantage of us as an animal model is because it's for the ethological aspect, it's really a perfect model for study behaviors, and it also is a good model for interrogation of potential therapeutic targets. For example, when we investigate this safety, the non-human primates may share similar innate immunity and immunogenicity. So, for example, if we are looking at the neutralizing antibody against the certain serotypes of the AAV, the mice won't be able to tell and use much of the useful information. And also, it's easier to study the situation when we need to scale up the AAVs and see their efficacies, such as looking at the peripheral vein delivery. The NHP will be a much better model, and also because they have a longer time lifespan, it's very suitable for the long-term follow-up for the efficacy and also for the safety. So when using the NHP animals, people might generate the disease model in NHP, and here are some examples. One of the study that they used AAV-mediated CRISPR-Cas9 to in situ knock out their target gene in the mouse eye to generate the disease model. And although doing the modify the target gene in the germline is also applicable, but using the AAV-mediated CRISPR-Cas9 system is a much faster and easier to operate system and can generate the disease model much faster than the germline. And there's also another approach for try to express the antibodies drugs into the patients via AAV, and which we call them AAV vectored immuno prophylaxis. So in that case, we will be able to replace the sequence within the ITR using the therapeutic antibodies, and we can, in that case, deliver the antibody into the patients in the long term without, repetitively dosing it as a protein format. So, generating these kind of models in the NHP can really mimic what we finally would like to do in human patients. Also, when doing the safety study, um, NHP has also a lot of advantages. For example, when we do the toxicology study uh, to study the neurodegenerative disease, it can be easily used to evaluate, for example, the dorsal retinal ganglions, and also to study the peripheral nerve toxicity, such as the hepto toxicity, and it can also study the adverse immunogenicity, the genomic interrogation, the genomic risks, and also is there any insertional mutagenesis will occur during that process. And also it will be able to study the safety doses with the pre-existing or treatment emergent anti-capsid antibodies. Some other animal studies conducted in the NHP model is, for example, we can do the persistence study to see AAV genome, um, mainly as the will they be able to exist mainly as the extra chromosome, and or will the AAV still integrated into the host genome in the long term span? And actually, I remember last year we see James Wilson published a two-paper study the long-term genome integration of the AAV in, by studying like many cases in the NHP model. And also, we can also use NHPs to study uh, the the surgical dosing procedure and the delivery devices system intended for clinical use. Well, of course, there are still a lot of challenges when we use an NHP disease model to study the AAV-based gene therapy. The most obvious one is that the animal accessibility, and like the monkeys, primates, they're much more expensive than mice. The animal cost is really something that we have to consider carefully, and also the low availability is also a big concern. It's not always easy to get a monkey whenever you want, and especially for the genetically modified ones. So we have to be very careful when we plan out the experiment and make sure that we, succeed every time that we can, not wasting all the efforts and the costs that we provided. And also, it needs a longer time to prep to get it ready. So you have to also arrange a lot of other experiments to make sure that all the AV materials are ready for the animal experiment. And this is very hard to coordinate among different parties. The second one is because NHP are much larger than mice, and they of course need large doses. Depends on whether you injected it in locally or you would like to do the IV. The quantity that you need for the AAV vector is usually around 10 to the 13th to 10 to the 15th. So for the 10 to the 15th, that's really large quantity, and that's also give you a lot of cost for to produce those AAVs. And also, um, the toxicity requirements are much higher because you increase the doses, of course the residual um, materials and also the contamination and endotoxin that you bring into the animals are also going higher. And uh, for some of the tissues, they could be very sensitive to endotoxin and some of the residues. So you have to be very careful about the quality of the AAV vector that you're going to inject into the animals. Um, anyway, the NHP do requires a lot of careful consideration, and we think that providing a high quality viral vector can really help to bypass all these kind of risks that involves with the NHP study, and you know, but on the market, it's not always easy to find someone who can always provide, you know, all these requirements that can that you know all the required standards for the AAV vectors that can be used in the NHP. Most of the cases, you might find some of the you know, the contamination is too high, the empty capsid rate is too high or quite inconsistent. So every time you try to generate some data, it's not always so consistent, and sometimes it might result into the failure of the study. So yeah, so we actually heard some of these voices from our customer, and we last year developed the new grade of AAV material for our scientists, so we hope that we can help them to solve all these problems. We used to be, you know, very proud of our AAV research grade that we produce. We have been providing that for many years, and because we've seen more and more people using the NHP model, so we generated a new grade that has the upgraded feature. And what we have done here is we will provide more control for the endotoxin, make sure that the all the materials was low endotoxin, and will not have you guys to worry about the safety issues. And also we will um, have more precisely quantify the materials to you. So if the quantification, like the titer of the AAV, is not very accurate, you might get different results, even if you try to, standardize all the conditions. You may not have your results very reproducible. So we heard your voice, and we will have all different methods to make sure that we measure the really effective titer very carefully, and try to avoid any inconsistencies between different batches. And of course, contamination is something that we always want to avoid. And for the NHP grade, we give much stringent control for the contamination, and we have all kind of options that we can provide. Um, so here is just our production process, how we generate the NHP grade AAV. Just like how we do our research grade, we start with from the vector construction, but not only we verify the gene of the interest sequence. In the NHP cases, we always verify multiple times of the ITR sequence, to make sure that the ITR is not mutated or deleted during the process. In that case, we will not see much of the variants when we do the viral packaging, and also make sure the genome being delivered is intact. And then after that, we will purify the plasmid. We will check the ITR again before we put into the packaging system. So when we fermented the packaging cell lines, we can have many different options. So we can use small systems, and we can also scale up easily using the fermenter, so to save the production cost. And then we will harvest these cells, and we will then go through multiple purifications. So the purification steps here is much more complicated than a research grade, so we can get rid of a lot of contaminations, and also get rid of the empty capsid. So we'll finally be able to get a very good quality of AAV. And of course, these AAV will go through the sterile filtration before we move on to the QC test and the QA review. So here is a comparison between the research grade and the NHP grade. And we do have several upgrades of our default QC. The first that I would like to mention is that the titration. For the research grade, we use qPCR to measure the titer to give you an estimation of the functional genomes that within the AAVs. In the NHP, we will use a much more precise measurements by the ddPCR. So I will tell the advantages of ddPCR in the later slides. Also, for the purity analysis, in the default format, we will still run the SDS page, but for the ANHP grade, we will do the silver staining, which will give you a much crisper images of the capsid. And then, of course, for the endotoxin, we will make sure the endotoxin is less than 1 EU per milliliter compared to less than 10 EU milliliter for the research grade. We will also do the bioburden. Of course, you can also switch to the sterile, which probably would take much longer, but you can also do that. And we do check the genome integrity to make sure that the genome we pack in the virus are most of the cases are intact instead of we are packaging the truncated genome. But this is also very important to know that the majority of the genomes are effective and will give you the best efficacy. Of course, empty capsids is something that we always like to control. NHP grade have some more stringent QC standards. For the NHP grade, we guarantee less than 20% of the empty capsid, and anything above that, we will not release our sample. And also, just rest assured, in most of the cases, 20% is we would consider it's not a very stringent stringent thing to pass. Most of the cases we can get around like 5%, even less than 1% of the empty capsid. So you can pretty much rest assured that the um, AAV that you get is having the intact genome packed inside, and we will show you the best efficacy. Um, so our NHP grade, in order to produce high quality, we do not produce it in our research lab, and instead we produce them in our GMP facility in our PD lab. And and there, because we have a lot of different fermenters, we can easily control the production scale in a cost-effective manner. And we can also use either the adherent cell line or the serum-free suspension culture, in case that you would like to, get the production conditions very similar to the later on GMP production. So that will be a very good transition when you are ready to get into the GMP production. And and also, when we produce NHP grade AAV, we use our high alpha 293 high yield AAV production platforms. So this platform consists of several different components. One of the key components is our proprietary RC plasmid that we modified in-house, and this has been registered for the patent. And this plasmid can help us to produce over 3 to 8 times more of the AAV compared to the unmodified ones. And we have tested over different projects with the different serotypes and shows very promising data. And also in the platforms, we use our own proprietary cell line, and um, we have the adherent cell lines, and we also have suspension cell lines that we keep evolving. And these two cell lines can also increase the yield several fold. So you can actually choose whether you want to produce your AAV in the adherent cell line or the suspension cell line. In case that you would like to get ready for the IND filing and ready to move on into the GMP production. And also the production data that in this preclinical stage can also be easily transit to the GMP scale later on. And also because the ANHP grade will comes with more stringent QC, and besides all the default QCs that we just talked about, we also have many different assays that's available for the releasing test. So this consists of over 60 already developed assays, and they have they are also converted from our GMP assays, so it's totally reliable. And later on, you can also refer to all these testings to develop your analytical panels for your GMP level of production. So here I would like to show you a few examples of the QC standards that the QC tests that we included or recommended for AAVs that are being used in the NHP study. The first, of course, is the ones that we highly recommended, and we included as our default QC is the ddPCR. So we found that the ddPCR has a lot of advantages over qPCRs, just because it definitely provide a much more accuracy compared to the qPCR. It's the absolute quantification instead of the non absolute qPCR, because it's for the qPCR it's more of the analog measurement, and it requires the standard curve. But for the ddPCR, it's really kind of a absolute value, and it does not require any standard curves, and it has way much higher sensitivity compared to qPCR. For qPCR, you probably only be able to distinguish less than two-fold copies of the numbers uh, for the expression variation. But for the ddPCR, the alleles that are less frequent than 0.1%, you will be able to tell. And also it's much more precise than qPCR, and definitely gives you a much better consistency batch over batch. The other task that we highly recommend, and we kind of included in the default QC, is of course the capsid characterization by using the SDS page. And here we use a silver stain to show a much crispier band. Um, and this is more, it's a very direct way to see whether the capsids are intact in your sample. And also besides that, we would like to make sure that the genomes being packed into these capsids are intact. So we recommend using CE to measure the genome integrity. And this is also very effective method. And you can easily quantify how much of the genomes within the population is intact, and is there anything that's kind of not intact or has a little bit, extra things being packed in there. And TEMs is also very popular methods, and here we're just having a much more higher, much more stringent standards for we can tolerate the empty capsid. Um, some other experiment that we do not include in the default package, but we highly recommend, is we would like to recommend the ones for measuring empty capsid by the analytical ultracentrifuge, and can be more quantifiable compared to the TEM, and for the analytical ultracentrifuge, it will be able to tell how much percentage of the capsid are full, and if there's any capsid that's aggregated, which will be kind of the toxic and generate some immunogenicity within the animal. And also it will be able to tell how much empty capsids was in there, which could lower the efficacy as well as bringing more adverse effects on the animals. Uh, Contamination is also something that we hate. And for the mycoplasma test that we use in the NHP, we definitely will recommend qPCR, which is much more sensitive compared to the ones by using the kit, and we can see that there's absolutely no mycoplasma in the sample before we add it into the animal. And also, we recommend the nanopore sequencing. So nowadays, people are concerning, you know, we would like to see the ITRs were intact, and our gene of interest is intact before we put into the packaging system. But how does it look like when we package into the virus, and are the the ITRs still intact? Or is there any additional mutations being introduced during that process, and that could arise some of the safety concerns. So here we have the ones we established the nanopore sequencing, which gives you a very deep reads, usually over 10,000 read depth. So you can easily tell if there's any mutations or deletions in there, and we will not only provide the alignment, the histogram, the mapping of the ITR to ITR region, but we will also give you the raw data for you to analysis in that. So beyond all these tests we just show there, I have given you a complete list of the analytical services that we have available, so you can always choose from different panels and see if there's anything that fits your specific needs. So all these tests can be categorized into the titration and the genome integrity, the capsid characterization, purity, and aggregation, contamination, and safety assays. And also, all of these assays have their GMP version, so later on you can easily switch to GMP version when you try to do your analytical development. Last, I would like to mention that the plasmid we put into the AAV packaging for the NHP grade is also very stringent. We strictly control the supercoil, the endotoxin, and also the ITR to ITR sequence. As well, we also have additional QCs available for monitoring the residual host proteins, residual RNA, pretty much all the QC standards that you can think of when you do the GMP level of the plasmid. Here's just the example of our ITR sequencing for the plasmid that we put into the virus packaging system, and you can see from the clone screening to the final scale up plasmid, everything was pretty clear, and the Sanger sequencing that we can read through the ITR shows the ITR is pretty intact. Finally, I would like to briefly introduce, how packaging developed in the recent years. So we start in 2014, and our main production site is in Guangzhou, China, and we have commercial sites in Europe as well as North America. And last year, we expanded our footage into Houston, Texas, to build our GMP AAV facility, and we are looking forward to its fully launch in H2 this year, and we can produce the GMP from a US site. And here also, I would like to briefly mention our breakthrough AAV technology. We will introduce them in detail in our next few webinars. So please stay tuned. So our platforms we are going to introduce, they include the 293 AAV high yield platform that we briefly mentioned here today, but we will talk more details in the next few webinars that are coming up. And we'll also talk about our Pi Omega plasmid DNA high yield production system, which consists of different fermentation and purification methods, the transfection reagents, and how it can bring down the cost for the AAV production. And we will also find another chance to introduce our Pi Ecosa AAV capsid engineering platform that can help you to generate novel capsid that direct your AAV to the specific target tissues. And finally, Here's just to show some brief information of our Houston site that still under construction. Our process development and analytical development labs is already in fully operation. And if you have any projects that we can probably start from the process development part. And in H2 this year, our GMP facility will fully launch in our Houston site. And if you have any questions, please feel free to contact us. Also, you can visit our website or call us. Our technical support team will always be able to assist you for your projects from the beginning to the end. And I really appreciate you to attend this webinar. And we are ready to take questions.
Hi, thank you, Irene, for the insightful webinar. And now let's move on to the Q&A session. We have received some questions from the audience already, and we do encourage you to keep them coming. Feel free to type in the ask a question section in the below, and let's move on with the first question we received. So Dr. Song, what kind of serum-free HEK cell culture media do you use?
Hello. Hi. Yeah. So thanks for the question, and I really appreciate everyone to attend the webinar. So for the mediums that we use for the serum-free culture, we actually use Optimum, but we are also actually kind of evolving our cell line, and which we are still adjusting the formulation of the medium. So later on, when we have the newer version of the cell line launched, then we might using our optimized formulations. Yeah.
Thank you for the answer. And let's move on to the next one. Could you please tell us more about the recap non-coding regulatory element?
Yeah, so actually, I probably won't be able to give much details about it. This is one of the proprietary elements that we developed in-house and for quite a couple of years. And this is actually a regulated element that inserted after the rapid cabin gene. And so this plasmid has already been filed with the patent, and it's included in the platform. So if you have any project that coming to us, just need us to produce the AAV, we don't charge additionally for using that that plasmid. Yeah, so I think like we can give more performance of this plasmid, but we probably won't be able to tell what's the sequence, what's the the feature of the elements in there.
Okay, thank you. So the third one I have here is how long does it take for packaging to make NHP grade AAV?
So for the NHP grade, usually, if in case that if we need to have a little bit larger scale, for example, like 10 to the 14th, we might need to do some small scale tests. That's why we usually takes about 3 to 4 weeks if we have the plasmid template, and we can start from there and making the virus delivered to you.
Okay, sounds great. And then the next one I have here is what are the purification methods used for NHP grade AAV?
So the purification steps that we have, using in NHPs, basically we have multiple rounds of, chromatography, ultracentrifuge, and also tangential flow filtration. But in the different combinations, so we can have the all the empty capsid being removed, and also a lot of the residuals, plasma residuals, histidine residuals being removed.
Okay. And then the next one, are there any FDA guidelines on how to choose model animals for non-clinical studies?
Yeah, so FDA actually published a series of guidelines for choosing, how do you conduct the non-clinical studies. And in those guidelines, there's also information about what you need to consider when you choose your model animals. There's a very good paper published on the let me see, from the human gene therapy in 2022 that remember the author is Jeffrey Moffat, and in his paper he clearly discussed like what kind of things that you need to consider based on the FDA guidelines, and mainly it talks about like you need to consider the translation of the dose response for the pharmacology and toxicology profiles to human beings, the impact of the disease, biodistribution, all the different aspects. And I strongly recommended that paper. So it will give you, a summarized information. And of course, he listed, all these different versions of a original FDA guideline in his paper.
Thank you. And then the next one, what assay is used to quantify AAV purity? Because in the presentation you stated that you guys guarantee a 90% purity.
I think you're probably talking about um, um, the ones you use for the genome integrity, right? The ones that shows like 97%. So we actually showed like two different graphs. One is like we measure the AAV genome integrity by looking at how much percent of the AAV has an intact genome, how much have either the truncated version that shows like a small split peak, and that one we kind of guarantee over our QC standards actually guarantees over 80% of the population that have intact genome, but most of the cases we can do a lot better. There's also another graph that shows that we have the full capsid. That one was measured by the analytical ultracentrifuge. So that one we can sort of measure like the full capsid. That's the ones who's gonna giving you the effectively deliver your gene interest. That one we only give you a report, but I would say most of the cases we will see over 80%, sometimes over 90% of the full capsid in the sample.
Okay, thank you so much for that answer. Next one I have here is which of the following is better, nanopore sequencing or Illumina or PacBio?
So I would say for looking at the genome integrity, and especially you want to know more details about the mutations, the truncations, how much population that I would personally prefer either nanopore or PacBio, the long reads, because they can easily read through the ITR from ITR to ITR region, and especially because the ITR is highly repetitive. Illumina short reads, they might easy to make some mistakes when you try to align all these different reads together. Sometimes it's a little bit challenging for us to align the sequence within the ITR. So you might miss some of the populations that have the truncated sequence. But nanopore and PacBio is something that easy to tell. And I would say currently nanopore method is probably more mature, probably because a couple of years ago, I believe Dr. Philip Tai from UMass, that he published a talk on ASGCT about how to establish the sequencing to investigate the AAV genome integrity from ITR to ITR, and that method has been adopted quite popular, and we are kind of also using that method to do that. And on the operation side, it's much easier to operate the nanopore because it's you don't have to accumulate a lot of samples to start the run, and you don't have to wait for a lot of samples to be loaded. So it's much easier to do that in-house. And it does have a lot of information to tell. Just the one thing that have to be keep in mind is nanopore do have higher error rate. So QC your data is also more important before you put all these reads together. Yeah.
Okay, thank you. And then the next one I have here is how do you remove the endotoxins?
Endotoxins, I would say for the AAVs, most of the cases when we do the production, like the facilities, if the facilities, the condition is good, we don't have to do additional endotoxin removal. But in cases that situation happens, we might use Triton to remove it. But of course, Triton will give some adverse effects on the animals. So we will also if in case that we need to do the endotoxin removal, we will also do another assays to check the residual Triton and make sure the Triton level is below the threshold to be used in the animal.
Okay, thank you. And then does AAV virus-induced immune response prevent from repeated injection of AAV virus?
Just so I think that I'm not sure if I understand the question clearly, but I think you're asking if the AAV virus induced immune response, and then next when you do the next doses, will that give you the you don't have any problem of the immune from the AAV, but that's actually the opposite way. I would say if the patients or the model animal already develop a certain AAV immunity against AAV, it's probably not a good situation, and the second dose is probably already have the antibodies in their in the patients or animal, and actually in that case you might have to either try a different serotype, the engineering serotype, to bypass that, or you might have to increase the doses. That's actually some of the drawbacks of the AAV therapy, and that's something that we have to do our best to avoid, and also something we would like to know possibly happens, so we can know the right dosage.
Okay, thank you. I think we have time for one more question. So do you have the denaturing and non-denaturing agarose gel for QC?
Yes, we do have the denatured and gel agarose for QC.
Okay, thank you so much. So I think that completes today's session, and I want to express our gratitude to our speaker and your active participation. The insights shared today will contribute to our collective knowledge, and don't forget to stay connected by following us on LinkedIn if there's any question, and feel free to email us too. On behalf of Packaging, I would like to thank you for joining us, and we look forward to seeing you in our future webinars. Have a great day.
Thank you.
Thank you. Bye-bye.
Bye-bye.